now to the African area, and I would like to ask Dr. Louis Roberts, who's a professor of gastroenterology from the Mayo Clinics, to come to the stage. And he will talk about the updates of Africa Hepatopancreatic Biliary Cancer Consortium and the Global CCA Genome-Wide Association Study. And may I say that I looked up that the reason why you do it from the Mayo Clinic is that you did your medical doctor in Ghana. Is that correct?
Yes.
Okay. Well, the stage is yours.
Great. Good afternoon. I'd like to thank the AMMF and Helen particularly for the invitation to present here. So I think I'll actually flip my talk, and I'll start with the Global Cholangiocarcinoma Genome-Wide Association Study and end with the update from the Africa Hepatopancreatic Biliary Cancer Consortium, and have some disclosures. So just sort of broadly, why do we focus on epidemiology and risk factors? Because prevention is always better than the cure. And why do we emphasize screening then and early cancer detection? Because early stage cancer is more easily curable. And why do we study tumor biology and the tumor microenvironment? Because every patient with advanced cancer that I have met wants to be an exceptional responder. So those are some of the principles that guide much of the work we do. And so one of the reasons we started this international consortium for the genetics of biliary tract cancers was the idea of better understanding the risk for cancer and to see if we could better understand inherited risk for biliary tract cancers, in this case particularly cholangiocarcinoma. And so the initial study that we did, and here are some of our collaborators here that we've worked together with, was to try to see if using a genome-wide approach, we can find new areas in the genome where there are variants in the genes that are associated with increased risk of bile duct cancer. So I'll just talk a little bit about exploring the genetics of cholangiocarcinoma briefly. I'll describe some of the different studies we do, the genome-wide association studies versus exome sequencing studies. And I'll introduce this study, which we really completed the discovery phase for, and several of the collaborators actually are here in this room who have contributed samples for that. And we have embarked now on an expansion phase where we are hoping to increase the size of the study. So in terms of risk factors, we've talked about this a fair bit about, for example, obesity, diabetes, hypertension, metabolic syndrome as a risk factor, and gallstones specifically. We talked about PSC and other autoimmune diseases. Those seem to work through mediating chronic inflammation in the body. But we've also talked about liver flukes. We talked about viral hepatitis. And we've talked about the environment as well, and the fact that the environment also potentially toxins and other exposures might contribute all to cholangiocarcinoma. And so there's multiple factors that we think contribute to it. And I put genetics up there because genetics presumably can influence some of these, the way that some of these other factors lead to inflammation, for example. So there have been no obvious strong genetic links for cholangiocarcinoma in the way that we've seen them for other diseases. Now, we've talked about the fact that some patients with cholangiocarcinoma have mismatch repair deficiency, for example, that's known to be associated, that has a genetic cause. But really, in terms of, if you look at families, for example, there's only a few familial clusters that have been identified where multiple family members got cholangiocarcinoma. And many of the links that have been studied are attributed to the genetics of the risk factors. So, for example, people who have higher risk of diabetes, sometimes there's a genetic component, or there might be a genetic component to development of gallstones, for example. On the other hand, if we look in the cholangiocarcinomas, we find that many of the mutations that are found in the cancers are seen in other types of cancers as well. So it's not like we see individual mutations in cholangiocarcinomas that are only seen in cholangiocarcinoma as well. So they seem to be common to other cancers as well. We've talked about the fact that the genetic profile of the tumors, though, is different between the intra and extrahepatic cholangiocarcinomas, and the risk factors, are broadly different. We tend to see more cirrhosis and viral hepatitis and so on, more associated with intrahepatic than extrahepatic cholangiocarcinoma. And then, but having said that, many of the genetic studies that have been done have been in relatively small numbers of patients. And so just thinking about our genome, so we have chromosomes in 23 pairs, and of course, these expand out to the single nucleotide level, where we see that you have individual nucleotides, and these nucleotides can be changed from what they are in most people. They can vary into a different nucleotide. And we call that a single nucleotide polymorphism, or a SNP. And of our total nucleotides, about 3.2 billion, in general, about only 0.4% of them vary a lot. And so there's only 4 to 5 million SNPs that are thought to be common, that you see in more than 1% of people. And then there's up to 80 million that are rare, that are seen in less than 1% of people. So variation occurs in some of our genome, but it's a relatively small proportion of our genome. And these genetic variants, or you can think of them as mutations, mostly occur normally. We see them about 1 in every 1,000 nucleotides. And as I mentioned before, there's about 4 to 5 million in any individual person's genome. Most of them are not disease-causing. But we know that variants within important parts of genes can affect the protein's function, and can also predispose people to cancer. And so in these genome-wide association studies, we study the DNA sequences to identify SNPs that are present at different frequencies in cases versus control, so in cancer versus people without cancer. And to look for common SNPs, we use genotyping SNPs that have been, in a sense, layered with multiple of the common SNPs, so that in one chip we can assay all of the common SNPs. So typically we can assay about 1 million or 1 million to 2 million SNPs. On the other hand, to look for a rare SNP, we have to sequence either all of the expressed regions of the genome, or what we call the exomes, from which DNA is transcribed into the mature messenger RNA. So we do exome sequencing, or we might more recently sequence the entire genome, as that has become less expensive. So we do whole genome sequencing. The challenge, though, with these types of studies is that we need to do it in very large numbers of individuals in order to confirm that any one change we see is statistically significant. So in our cases with the cholangiocarcinoma patients, and as we've been talking, in general, cholangiocarcinoma is less frequent than other cancers, so it's more difficult to find patients with cholangiocarcinoma. So once we do these analyses, how do we represent the results? We do what they call, and has been referred to as a Manhattan plot, and the idea is that what the plot looks like is a little bit reflective of the Manhattan skyline. And so basically what we do is we do a series of comparisons. We look at any one of these SNPs, and we look at how frequently we see it in affected cases and compare it to unaffected controls. So in this case, in affected cases, it was 41%. It was 13% in unaffected controls. We would do a statistical test called the t-test, and we would plot the value of this, the p-value of that test on this scale, where it's the negative log, and so a small p-value is actually up here, and a high p-value, a large p-value is here. So things that are more significant will be a higher number. And so it's a little bit complicated because we do so many of these comparisons. We have to adjust for the comparisons because we have a problem of what they call in statistics multiple comparisons. And this is the statistician that is responsible for the commonest method that we use to do these corrections. We basically, in his Bonferroni correction, we divide the p-value that we got by the number of tests that we did, the number of SNPs. So we actually have to have a really, really small p-value in order to think that a specific change we find is significant. So having said that, for this study, we gathered samples from a number of different countries. You can see the United States, Canada, mostly European countries, and then Australia. And for cholangiocarcinoma, we're able to assemble about 2,600 patient samples. For gallbladder cancer, we assemble about 600. And I'll be telling you primarily about the cholangiocarcinoma study here. And so these were mostly people of European ancestry, and a total number of cholangiocarcinomas of people of European ancestry was about 2,400. And breaking it down by the subtypes, we had about 900 intrahepatic, 1,000 extrahepatic, PSC associated about 200 of them, and then you had the other gallbladders, and we broke back out non-Hispanics versus Hispanics as well. So this is the initial discovery phase results. This is looking at people of European ancestries, all of the cholangiocarcinomas. So to see if we take all cholangiocarcinomas, do we see variants that are associated with them? And we use about 3 times the number of controls as we had cases. And so this is looking at, again, the plots of the p-values. This is the chromosome, so starting at chromosome 1, and we start at the beginning to the end of that chromosome, and then we do 2 and on the way to 21, and then the X and Y chromosomes. And so what you'll see here is that there's one region here on chromosome 6 that is within the HLA region that's associated with a lot of our immune responses that is quite high here. And so this is a major peak. Usually the cutoff for significance is 5 times 10 to the minus 8 for the p-value. And you can see that this really, in this all cholangiocarcinoma, this was the only one that reached statistical significance. But there's a couple that are very suggestive because they are above this 10 to the minus 5 level that's used for suggestive or suspicious. And you can see here that there's a region in chromosome 5 that is significant around this gene, the CLPTM1L, and then chromosome 12, this LGR5, also suggestive. And there's other areas that are suggestive as well, which is why we are wanting to do the larger expansion phase, because the statistical genetics tell us when they see this, you just need more samples. So if we looked at intrahepatic cholangiocarcinoma, then again, we see nothing that reaches statistical significance, but several areas that are highly suggestive as well. And then I'll go to the extrahepatic cholangiocarcinomas. And here again, there's a different region in the HLA region that comes up. And then this is within a long non-coding RNA and a region that also is significant. So these are some of the ALA results that we found. And as I mentioned, we're currently undergoing an expansion phase. We're fortunate to be awarded a grant from the National Cancer Institute. And our goal is to assemble and sequence an additional 7,000 cases and controls over a 5-year period. We have a number of controls that we've hoping to collaborate with. Our emphasis is on global representation to try to understand more about cholangiocarcinoma around the world. And we've already actually sent a first batch of cases and controls for the expansion sequencing. So hopefully there's a map of some of the different sites, and we are trying to assemble sites, particularly in South America and Africa. There have been some studies done already in Asia, and we're hoping that we can fill out the global landscape of cholangiocarcinoma. So I'll go on. So if you have any interest in collaborating, please contact me. We'd be very happy to include you in the collaborations. So I'll talk now, the last few minutes, about the Africa Hepatopancreatibiliary Cancer Consortium. And this is also a special interest group of the Africa Organization for Research and Training in Cancer, which is an organization that seeks to improve cancer research, cancer training, cancer care broadly across Africa. And so the origins really are that I started out after, I completed my medical training in Ghana, which is where I grew up, but was always very interested in research. And so in medical school, one of my professors stopped me in the hallway and said, you're so inquisitive about things, you should really consider getting a PhD when you're done. And so that led me, when I finished medical school, to the United States. So I completed a PhD and then completed my clinical training. I was always very interested in helping with medical education in Ghana, so I began collaborating with colleagues and developing continuing medical education programs in sub-Saharan Africa. And in the course of that, recognized unique things about the burden of gastrointestinal and liver diseases in Africa, particularly, I would say, the fact that yes, we recognize there's a fairly high rate of viral hepatitis and liver cancer in sub-Saharan Africa, but it was interesting to see that people were seeing liver cancer at really young ages. So we're having the conversation that you've been having today about young age of onset of cholangiocarcinoma in Africa. And we're trying to figure out, what do we do about this? And it was important, we figured, to try to understand it better. And so we began advocating, of course, for the things we talked about: prevention, early detection, and treatment of liver, pancreas, and biliary tract cancers in Africa. And I won't go through this, but this is just some of my path to some of the networks we developed. The first network we developed was this network for liver digestive diseases. And that led us to develop, we did a workshop on developing liver disease registries in West Africa, which led to this publication, where in 7 countries across Africa, we showed that the median age at which people develop liver cancer is about 40 years. And so a lot of years of life lost in Africa from that. And so following on of that, several colleagues and I registered a nonprofit foundation in the United States. We called it the West Africa Institute for Liver Digestive Diseases, and then we very quickly realized that it wasn't just West Africa that had the need, so now we call it the Africa Institute for Liver and Digestive Diseases. And we then subsequently launched the Africa Hepatopancreatibiliary Cancer Consortium, and started with a series of conferences. And I'll just summarize objectives to say that we're really interested in understanding these cancers better, so research. We're interested in helping with training of both researchers and healthcare workers in terms of just improving care. And the third thing, of course, is improving early detection and treatment of these cancers. So I'll skip a couple of these and say we have an executive committee, we have a number of working groups, basic and translational sciences, epidemiology and population-based engagement, and clinical training and care, and then subcommittees that helped really to support these efforts. And I'll skip here. We started a series of conferences. We're fortunate to get some support for these. And so we held the first one in Cairo in Egypt in 2022, had about 160 attendees from about 15 countries. Then in 2023, had a conference in Ghana that had about, 200 attendees from 20 countries. And then this last conference last year in Mombasa in Kenya had about 260 attendees from about 29 different countries. So we're finding a lot of interest from our colleagues in Africa in learning more about these diseases and trying to treat patients better. We do a lot of pre-conference hands-on workshops, and you can see we're getting attendance from lots of different countries. And here's a typical conference, some of the different groups that we formed. Advanced endoscopy, because in many parts of the continent, there's no way of diagnosing cholangiocarcinoma because you can't do an ERCP. Pathology, ultrasound for the screening, and then patient advocacy as well. And we do these clinical skills workshops as well, do a surgical workshop, do endoscopy workshops. And I mentioned the advanced endoscopy training particularly as a key with our interest in helping with both cholangiocarcinoma and pancreas cancer. We started a biorepository effort because we realized that it's important to have samples from these countries in order to be able to study them and better understand the biology of the cancers. And then we are helping in different countries in different ways. For example, in Nigeria, they've identified liver transplantation as a key need and don't have any liver transplant program. So we're working with colleagues on the continent to help them develop a transplant program. So our next meeting will be in August, from August 12th to 16th in Johannesburg in South Africa. So you're very welcome to consider attending it or supporting it. We have a webinar series, and some of the topics have been leveraging genomics in cancer and research, or the role of proteomics in cancer research. And so we have an active series of webinars. We've tried to have research awards, and we try to particularly train individuals in grant writing so that we can increase the numbers of young scientists in Africa that are doing work in Africa. Dr. Nate is from Ghana, Dr. Zaki from Egypt, and Dr. Kampore from Burkina Faso, for example. And just an example project that we're doing, this is a prevention project. It's called Stop Hepatitis C in Ghana. And it's an interesting project. It actually got featured in the New York Times a couple of years ago because Egypt had very high rates of hepatitis C and did a huge national campaign for the eradication of hepatitis C. They were able to get the free licenses to manufacture hepatitis C and the antivirals. Have two companies that manufactured the drugs. They screened over 60 million adolescents and adults and cured over 4 million people of hepatitis C. And then they made the announcement that they wanted to donate enough drug to treat 1 million people in sub-Saharan Africa. So working with the Ghana Health Service, we applied to the government of Egypt. They made a commitment to send enough drug to treat 50,000 people in Ghana, which would be about 1/10 of all of the cases we think that there are in Ghana. So we started this national hepatitis C screening campaign and working towards elimination of hepatitis C in Ghana. And there's the Egyptian ambassador at the meeting a couple of years ago where they made the first donation of these drugs in Ghana. And these are the treatment centers that we've developed in Ghana across the country. And we started about a year ago doing this nationwide campaign. And there's healthcare workers, there's the comedians in the country, there's the people who've been treated that can give the testimonials about the success of the treatment. And there's Easter eggs here, so if anybody wants to take a picture of these, there's a series of Instagram and Twitter messages that relate to the screening and treatment program. So I'll stop here, and I think that's how we can go on. Thank you.